Well, welcome everyone. What a great group. It's nice and cozy in here. This is wonderful. Thank you all for being here this evening. My name is Jerry Bowers, and I am the VP of External Affairs at the MDI Biolab, and I am thrilled to be here with my colleagues from the Bigelow Laboratory as well as the MDI Biological Lab. I know no one ever wants to sit in the front row, but these are cushy seats. We put the most comfortable seats up here to try to entice you. So we are really, really pleased to be here this evening at Stone Coast Fund Services. This is our second Science Cafe that we have done here in Portland, and are so grateful to our hosts, Mark Kepper and Kenyon Schubert, who have just done a wonderful job of making us feel so at home here. And so I hope you enjoy this beautiful location. And even more so, we are super excited to tell you a little bit about a new collaboration between these two research institutions here in Maine. So we're very excited to share some details. And I think this is really the first time that we've talked about it publicly. So I think there'll be a press release coming shortly. But tonight we wanted to give you a chance to hear from our scientists and our collaborators in terms of what this project is all about and how it, we think, may really move Maine's R&D sector forward. So before we begin, I want to just set the stage for you a little bit. I don't know if any of you have participated in science cafes before. This is something that I think both of our institutions do, and they are an international movement. They're really designed to be a conversation, so not PowerPoint, right? We're moving away from the lecture style, and so you may not realize it, but your job, you actually have a homework assignment here tonight, is to really ask some questions. We want this to be interactive and collaborative, and so we want to make sure that the information we're sharing with you this evening is clear and easy to understand, and so we look forward to having your participation in this conversation. But to begin, you know, for me, I grew up here in Maine. I'm a 9th generation native, and so it's just such an exciting time in the R&D sector, the life science sector in particular here in Maine. It's something that I think as a sector, we've worked really hard, and Agnieszka from Biomedical has been one of the folks who's really led that charge to try and shine a spotlight on the science activity that's happening here in the state. And, you know, we tend to think of ourselves as kind of fishing, farming, and forestry, but in reality, there's a lot of science that goes into all of those things. And so we'd like to add some biotechnology to that, to the main shtick. And so we are excited to talk a little bit about that tonight. And interestingly enough, you know, the main biotech sector and life sciences in particular has grown about 42% over the last 5 years. So that's stronger job growth than any other sector in the state of Maine, and actually the fastest growing sector in New England. So I think this is, you know, a really important message that Maine is poised for some exciting growth in the future. And we think that this particular collaboration is a great example of how the sector is growing and how these opportunities are really presenting themselves. Despite that, you know, Maine still continues to lag nationally in terms of where we are with R&D activity. So there's a lot of work to do, something that we talk a lot about with our legislators, and, you know, trying to continually talk about the need for support for science in Maine. But with that, I want to just introduce our panelists this evening and tell you a little bit about each of them and what they bring to this conversation. So to my right immediately is Jim Strickland, who is he is the Senior Director of Strategic Initiatives and Biotechnology. He's also the Director of MDI Bioscience, and he's going to tell you a little bit more about that particular division of the MDI Biological Lab. We have Dr. Deb Bronk, who is the Senior Research Scientist and CEO of the Bigelow Laboratory for Ocean Sciences. And beside her is Dr. Herman Haller, who is the President of the MDI Biological Laboratory. And then Dr. Beth Orchid, who is the VP of Research, a Senior Research Scientist at Bigelow. So welcome. Thank you for being here. And this evening, we're going to talk a little bit about something called bioprospecting. Jim and I were talking about this term, this idea of looking to nature for potential therapeutics. And so what I'd love to do is have just our panelists first just introduce themselves and talk a little bit about your role at your institution and kind of how lay the stage for our audience in terms of what your role is at the MDI Bio. I'll start with you, Jim.
Thank you, Jerry. So my role at MDIBL is really an exciting one, which is MDIBL science has been well known for 125 years. But in the last couple of years, we've really taken a new twist on the science and tried to also create a translational arm. And what that means is, how do you take that wonderful science and create applied uses for it that not only create opportunities to influence people's lives, but also create revenue streams for the organization. And so through that, we started MDI Bioscience, which is a new initiative that brings more of a translational focus to the lab and lab's work. And so I'm excited to be here as part of this project, which will be a central part of the collaboration.
Hi, I'm Deborah Bronk. So I'm the president and CEO for Bigelow Laboratory. So I guess I'm where the buck stops at the laboratory. The Bigelow has been around we're kind of a baby. We're so proud we're going to be 50 years old, but compared to MDIBL, we're a teenager. We'll be celebrating 50 years next year. Our kind of bread and butter and what we're known for internationally is the research. So the foundational research that's just trying to push the envelope on how the ocean works. And our area of focus is we study microbes. So we study the base of the ocean food web. So that's the microscopic phytoplankton, as well as the bacteria and viruses that exist within that community. We also study things like coral and kelp, which are the foundational species where they exist. And then we go one level up and we study the things that eat those things. And we're really quite focused in that. And I think within that space, I don't think there's a better lab in the world doing what we do. Recently, though, well, we've always done education. Our education programs are very much about immersing students in the labs. They become part of the research team. So we're never going to have 10,000 students coming through the lab. But if you want to launch a career in science or you want to figure out whether you want to be a scientist, we're a great place to come. What is fairly new in the last decade and where this collaboration comes in is into more research with a commercial application. So, and that comes, I think the interest there is twofold. One is to diversify our funding stream, but the second is because the scientists, so I've, for 30 years, I've done research. I'm an oceanographer by training and just watched climate change be a disaster that is slowly picking up speed. And I think there's a real urgency in a number of our scientists. And what we realize is that understanding a problem is just the first step. You've got to then get it into a solution. You got to get it into a product or a process that you can get into the market at scale. So over the last decade, that's one part of what we've been doing which took kind of a cultural shift within the laboratory to be able to do that. But partnerships are a big part of it. So we're thrilled with the partnership with MDIBL.
Thank you. So MDIBL, near Bar Harbor, far away from here. So what have these guys been doing over the last 125 years? Research, but what type of research? So what we are famous for is comparative research. In the old days, we compared different species from the ocean. This is where it all started. And then we found that each problem has its own animal. And when you compare these animals, you can really understand what's going on. We are also medical. I'm a medical doctor, so I'm interested in disease and I'm interested in how organs function. So this is what MDI-BL has been doing, investigating comparative research in animals in order to understand organ function. And for some organs, we are really world famous, for instance, for the kidney, but now we are also looking into other organs like the brain, we're looking into eyes, retina, and we have changed our animals. In the old days, it was the ocean, and now we have what we call non-mammalian animal species, which we can genetically alter. So I can't help it, you have to know that. We are doing C. elegans, which is a small roundworm, but one of the models for genetic research. We're doing zebrafish, swimming not in the ocean, but in the aquarium, and we can investigate these animals and we can genetically alter them. We do axolotl, we do a fish from Africa, African turquoise killifish, and last but not least, we do Drosophila. So we are one of the few labs worldwide where all these animals are looked at, and we do comparative research in one institution. Very nicely, we have on the island the other big lab, which is doing the mouse, which is the mammalian genetic animal model. So all of a sudden, Mount Desert Island is the center of comparative research, not only here in Maine, but internationally. So this is what we're doing. It's research orientated in basic research, and our researchers like yours are very proud that they are basic researchers. And now comes MDI Biosciences. So we have told them your wonderful animal models, our wonderful animal models can be used for biomedical purposes because we can genetically influence them, and then they are small and they are transparent. So we can actually look at different organs with our advanced microscopes. So this is our expertise for research, and now we're using this for biomedical and for pharmaceutical purposes. And now all of a sudden, we have this wonderful collaboration where these things come together and we can use this. It's really ideal. If I would have thought about it, we could have done a movie together.
We may still.
Good. Hi, everybody. My name's Beth Orcutt. I'm the relatively new Vice President for Research at Bigelow Lab. I've been there for 11 years as a senior research scientist myself, studying all the weird life that lives at the bottom of the ocean. But what I'm excited about doing at Bigelow is, as Debbie was talking about, really thinking about how we can take the expertise that we have to develop solutions for some of the world's biggest problems, whether that be climate change, ocean acidification, and not just thinking in the ocean, but how our expertise with ocean organisms can be applied to land-based problems, human health type problems. You may not know it, but the Bigelow Laboratory is the home to the world's most diverse collection of algae in the world. We have over...
Usually when we say that, people go, whoo. Okay, you can try that again.
World's biggest collection of algae. Okay. Most diverse collection. And so we are excited. That became part of Bigelow way back. We've had this culture collection for quite a long time. And our scientists are experts in growing all these different algae. They have all kinds of different needs. And they make different products. And so we knew that we had opportunities to think about how the algae in our collection could be used to find solutions. And so this new collaboration will be one of the ways that we try to look at that and think about human health and drug discovery type aspects, from ecological aspects, as well as the ways that we try to grow algae for solving other problems like nutrient capture, other types of products that algae may use. So we're very excited to bring our different institutes' expertise together and bring the scientists and all their enthusiasm to try to tackle these problems.
That's great. Thank you so much. So I'm sure you all can see where this is going, right? So we have experts who focus on human health, who utilize fish and other species that have great capacity for regeneration. But we have this wealth of expertise. We have scientists who have been studying various organisms like this for more than a century, so 125 years. And then you have the Bigelow that has this amazing collection of algae and microbiota. So we are very curious to see what happens when you put these two groups of expertise together. And so, Jim, Dr. Haller mentioned a little bit about MDI Bioscience and kind of, this is a relatively new initiative at MDIBL, kind of harnessing this expertise. Can you tell us a little bit about what the intent is and maybe your interest in, everybody's going to say it with me, right? The National Center for Marine Algae and Microbiota, the largest world's, center, largest collection. Woo-hoo. There we go. Right. Sorry, that was poorly done. But anyway, get the message. There you go. So you can just tell us a little bit, lay the stage for this.
Sure. So I think this is where I'm supposed to say bioprospecting.
Yes.
What is really at the heart of MDI Bioscience is discovering new cures and new ways to treat conditions that are previously untreatable. And so we know that marine organisms have incredible capacity to potentially influence disease processes based on a whole bunch of factors, which I'll let my colleagues at Bigelow talk to. But I think from MDI Bioscience's perspective, this is an enormous opportunity to use our core skills at investigating the mechanisms of disease, injury, and also how to rehabilitate injury. And bring them to the forefront of this project in a very central way where we can use the marine organisms to then essentially show that we can restore function to different disease processes. And the zebrafish in particular is a wonderful model for evaluating drugs, because the way drugs are typically discovered today usually involves starting with cells, and cells are great, but they don't tell you the whole story. And so a zebrafish is really a wonderful way to get the entire biological organism intact and show that you can have an influence on not only a particular cellular or molecular function, but you can also have the ability to study that and its entire influence on the organism itself. So it's really the first opportunity to show that you can demonstrably change the welfare of an organism. And so that tool gives us a tremendous flexibility to look into things like algae and microbiota, and to not only look for particular diseases, but also look at all those unexpected things that you can only see in an entire whole organism. And so from that perspective, we feel like we really are excited about this project, because marine algae and microbiota have the potential to be anti-inflammatory. They have the anti-cancer properties. They have neurological properties. And so all of these different potential applications can be evaluated in one organism at one time. And that's particularly exciting. And for MDI Bioscience to take this as a project is really exciting. We're really pleased to be a part of it.
That's great. So Beth, from your perspective, or Deb, either one, but tell us a little bit about sort of how you see this process going from your end. Are there particular sort of techniques or resources that you all will bring to this project? So Jim laid out for us kind of the intent is to really look for these potential therapeutics or nutritional supplements, whatever that is. What will that look like on your side? How will that work?
Yeah, so if you've never been to Bigelow, let me paint a picture for you. We have a state-of-the-art research lab on the Damariscotta River, And as part of our campus, we have a greenhouse where we can grow algae at scale. So like big vats of algae. But we also can do that in the laboratory at smaller scales, grow them in a flask, things like that. And we, as I mentioned, right, we have this incredibly diverse collection of algae and microbiota, bacteria, that we keep either cryopreserved, or some of them we keep them alive, right? We propagate them. And so our team there in the culture collection, right, they'll use their expertise of knowing all these different algae and how to grow them to as a service that we provide to customers all over the world, right? Scientists, nonprofits, corporations, right? They can request these algae from us. And so in the same way that we would grow algae for anyone, we will grow up specific algae for this collaboration, share them with the MDI-BL team to screen them, and I'll let you guys talk about that part, to see if the properties that we're interested in are detected. And depending on how that works, right, we might grow more algae, we might try different species, depending on what it is that the collaborative project is trying to look at. So there's a variety of scientists there that are really experts in trying to grow these algae. They're like algae whisperers, if you will, because I'm not good at that. I think one of the cool things about this project, right, is like we've got this in-house expertise in a culture collection in Maine that you don't have anywhere else in this country, and then this expertise with you guys. So I think, we don't have to ship it very far.
Yeah.
And one way to think about algae is the genetic potential in just a drop of water. When you think of all the different bacteria and microbes that are in there, it's enormous. And so, algae, if you look at the diagrams of all the different metabolic processes they do, it's crazily complex. So there's almost unlimited potential on what you could use these cells to do. If you have a knowledge of what they are, growing them under different conditions, they produce different substances. But we really were kind of stuck in terms of, okay, well, that's great. What does that mean? How do we translate that? And so that's where, I was bouncing off the walls when Jim came to give a talk, and I was like, this is how we do that. And so, it'll be an iterative process of, did this show any promise? Yes, but not great. Well, okay, well, there's 9 other species that are similar that do anyway. So we'll go back and forth, but think of algae as like these little factories that, depending on how you grow them, can pump out very different compounds, or the whole cell themselves can have like very different nutritional value.
So Dr. Haller, we have this amazing sort of resources on both sides, right? You set the stage for a great collaboration. How do you know where to start? What do you choose? Where do you begin?
Yeah, so I'll explain very briefly how we're going to do this, but the question is where to start. I think we're all convinced, and you are convinced, that's the two of you, but basically we all know algae are positive. When you think about algae, and you think about, would I use some cream for my skin? Yes, it's positive. Well, are there anti-cancer possibilities? Is it anti-inflammatory? Do you want to eat yogurt and algae and feel great for the rest of the day? So I think we all have a basic understanding, yes, this is positive. And now we are trying to find which substances. And how do we do this? So you'll talk about extraction processes and how to get the stuff out of the algae. But then Jim will receive, together with Sadie, some flasks, and they contain the secret of the deep ocean from the algae. And now the question is, what do we do with it? So we have these animals, and we have looked into our animals, and we will use zebrafish, and Sadie is working on using zebrafish embryos. They are very small, but they're transparent, but they have everything. They have kidneys which look like ours. They have hearts which are beating, they have a brain, so they are, as Jim already described, they are living small organisms. Now, how do we use them to screen for substances? First, we have to give them not a disease, but a condition we are interested in, and that's inflammation. So we expose these small, tiny fish, not to a large extent, it's not harmful, but we expose them to a substance which is called LPS, which makes them inflammatory. So yes, they're all inflammatory, but can we see that? You know, I'm a medical doctor, but I can't diagnose this. So I need a diagnostic tool, and the diagnostic tool is a gene which we have introduced and altered it so it's lighting up when they are inflamed. So these fish actually not only change their color, but they emit a fluorescent signal when they have inflammation going on.
So is that an inflamed fish?
That's not an inflamed fish, but this is a genetically altered zebrafish where the green stuff are molecules which have been labeled with green fluorescent protein, so you can see it. And we are experts in that. So this is why we can make inflammatory molecules. The molecule is called NF-kappa B, and you don't want to remember that, but that's a key element in inflammation, and then it lights up, and it lights up in the whole fish. So we can see this relatively easy. And now we have these fish, and when we get your extracts, we then start putting these extracts onto our fish, and when the fish lose their color, we have an anti-inflammatory effect. We will do this in the future for cancer, we will do this for others, but we have, after lengthy discussions, we have started because we want to have the aspirin of the next century, or the ibuprofen of the next century, or perhaps me as a nephrologist, an ibuprofen without any side effects on the kidney.
Mm-hmm.
Wouldn't that be something? And this is what we can make out of this. We are producing these fish, Sadie, if you work hard enough in your team, and then we have these fish, and if we get then the extracts, then the rest of it is a hell of a lot of work. You know, because how many substances do we expect to be in there? I mean, we have the first extract, we are talking about 100,000 of different molecules in there. And Jerry, just give me one example, because we discussed this on the way. Jerry asked me, so are there other examples? There are wonderful examples out there. In transplantation, I'm a transplant doctor, we have cyclosporin. And cyclosporin was introduced end of the 80s and has changed the whole field of transplantation worldwide. And it's from a plant on the Norwegian fjell. One of the researchers, the biologist from Novartis, came by and scratched the rock and had a little piece of the plant, and they extracted the drug from there. So you can imagine this gives us great hopes, really, to change not only our two institutions with that, but to have a huge impact in changing the world from Maine.
That's really exciting. So are there questions? I think we've laid out for you kind of the general, yes, there's one in the back. Congratulations.
Yes, it's super exciting. So bioprospecting, can you give a thought, or do you have an approach that says, we want to collaborate with existing, like gut biome out there, or cell therapy companies that are doing this to help them? Or if no, we want to go very novel new things that would help them too, or both, or maybe one of them?
At least initially, we're gonna see what we can find and pursue this on our own. But I think it could very quickly lead to partnerships with potential companies that are doing novel work in the areas that we're trying to pursue. But you know, just inflammation itself lends itself to so many different conditions, whether it's neurodegeneration or heart disease or kidney disease, or so it's a great place to start, and I think it'll lead us quickly to partnerships and other avenues to explore this project.
Great. Other questions? Yes.
Well, since I'm an attorney, I have to ask this question. How do you keep this proprietary to the organization? Are you an open source kind of institution? You publish your research. If you do, then how come those venture capital holders are out there kind of snag not only your discoveries, but your people? How do you prevent that?
Well, the secret's in the data, and the data don't have to be public unless we make them public. So from that perspective, I think we'll keep close tabs on our discoveries and make sure that we're protecting them to the best of our ability before we, as a team, make any decisions about what to do next. But
So our life has changed, I mean, really, science is a little bit different. We still try to keep science as open as possible at MDIBL. This is what we're known for. I mean, the freedom of science. And however, these days, before you open your mouth outside of MDIBL, you better talk to Tim about patent and could this be patent before? And it has changed us. I mean, the scientists do this. Not all of them, but most of them try to talk about that. And I've started 5 companies during my time, and I can tell you, I have learned this the hard way. What happens if you talk too early about your results?
So when I mentioned that going into the more commercial areas with research, there was a culture shift that had to happen at the lab, and that's it, right? So, we're chatty Cathys, right? We are scientists, we go, we give talks, we want to get the word out, and you had to learn what to do, what you can talk about, what you can't. The way I operate is with these kinds of partnerships, it's like, tell me what I can talk about, because I don't want to be in front of a large crowd and have to remember what I can say and not say. And when Beth heard about this event, she goes, is it wise that we should go and talk in front of a bunch of people about this? So it is, we've got to be careful, right? So we talk at a very high level, and then the scientists have to be careful, but it's a culture shift for the scientists.
But as you know, I mean, a group like this, that's enough. If you would name a molecule, which we haven't yet, we wouldn't do that. But to talk about this in general, no problem.
And maybe I'll add just one more thing. You talked about, our worry about people coming and poaching from us. I think it's actually the opposite, that we do want to be open about discussing what we're trying to do for the state of Maine and this blue economy, and right, to actually attract talent the other direction, right? And to show that there is cutting edge biotech research happening in Maine. Right, we see it as one of the great growth sectors for our state. Looking over here at Maine representative. So it's not about being quiet. It's about being exciting and attracting that talent to us.
And controlling the flow of information. And at least in Bigelow, so this is just one project we've got like this. I don't know if any of our scientists really want to go out and launch a company. Right. So if we get it to a certain point, we will be looking for partners that would then take it to the next level or to license it with a company. So we want those people to come and poach. The other thing is scientists, we could have gotten other jobs to make a lot more money if we wanted to in most cases, but they love the science. And so we have a lot of scientists at Bigelow have no interest in any of this, right? They want to stick with the basic research.
Patty?
So you talked about being careful in the media. So I read an article last week, I believe it was, where there was a South Australian research lab that was using blue-green algae, and they did a process to it, put a thin coating for wound care. So if you read that in the media, is that something like you pick up the phone and say, hey, what are you doing there? So is it a broader collaboration as well, or do you pretty much just stick to like, this is what we're doing?
One of our largest projects at Bigelow right now is we're developing a supplement to reduce methane production in cow rumen, because methane from cows is one of the main sources of a really harmful greenhouse gas. And it was actually a paper published in Australia that showed if you feed kelp to cows. So yeah, I don't know if Nicole called those researchers, but she definitely read the paper, and that led to a whole nother, I think it's our largest project right now.
Yes.
I have a question about what your vision is for this collaboration and how it can impact the workforce Maine for biosciences. Will it exist as a separate entity or center that kind of is populated by scientists from both of your institutions, or will you be drawing from the very busy already full-time jobs of your respective senior scientists? I'm just curious logistically how this collaboration plays out.
That's a great question, and definitely one that I had jotted down too. So I know, as Deb mentioned at the beginning, both institutions have a deep, deep history of training scientists, of education and hands-on research training. But I know from MDIBL's perspective, we've been talking a lot about sort of where some of the current maybe deficiencies are in workforce. And Jim, I know you and Dr. Haller have been thinking a lot about that. Do you want to talk a little bit about sort of where you see the potential for this going forward on the entrepreneurial side?
Sure. So great question. And I think the project as it stands now is really in a pilot phase. And so to the extent that we're at the beginning of the process, I think all options are on the table, and we're the great thing about a good partnership is we're all talking about it actively and trying to figure out where it goes. But I think the potential to spin out startups and really become an engine for economic development from a startup activity perspective is there. And then I think it also sort of articulates what in my mind is sort of a missing piece in Maine that workforce development could benefit from, which is translational science. How do we pull more of these technologies out of Bigelow and out of MDIBL so that they become technologies that ultimately can benefit the Maine ecosystem and help us as a state come together and really embrace a new cadre of companies and organizations that benefit from this type of translational work. And so that's a different kind of mindset. It's a different skill set, as we were talking about earlier. To be able to talk across business and science is a challenge. And so how do you build the process that puts everybody on a level playing field and establishes a language that everybody can speak about how that process of moving a technology from the bench to, in this case, the bedside happens. And I think that's a skill that I think I don't want to say more sophisticated. The Bostons and the San Diegos and the other places where biotechnology is spinning out regularly, that's a skill set that they have that I think we can learn from and sort of build in Maine.
From my side, in a concrete manner, we isolate, we take these specimens, we look, and then we try to isolate molecules. Each of these molecules, when they have a therapeutic or biomedical effect, is then a single company. A spin-off can only deal with one compound. You can't do 2 or 3 different when you are small. So each of the compounds, and here the imagination can run wild, is really, but I'm serious about that. Either you patent it and throw it away, or you can start a company. And Jim just mentioned this, and we are already thinking about that. So what do you need when you start a spin-off company? You need young people or people to take over. And this is what we lack in there. So we are already early on, not about this project, but in general, we're talking to our graduate students, what do you actually want to do as a career? And when they are more interested in the biotech area, we take this seriously. We educate them, we make courses so that they understand how to write a business plan besides their research project they are doing. And when you think this through, then all of a sudden, you need not only a CEO for such a company, but you need somebody who is doing the science and somebody who's doing the business. And all of a sudden, you're talking 5, 7, 8 people. And at the moment, to be honest, you can't find these people. If you want to start a biotech company in Maine today, you have a very hard time to find 8 trained people in these different areas. So 2 things come together. new spin-offs because of the compounds we will find, and then training of the young people to do that. And I fully understand that a lot of researchers are not interested in starting a company. When you are 24, I think you're more interested.
Well, but to start a company, it's a very different skill set than most scientists have, right? And so and the biotech ecosystem in Boston is really great at that. You know, there's a postdoc in the lab of one of the scientists that develops something, and then they take the compound and run with it. But you then need different partners, right? You need business partners, and you need, in our case, you'd probably need organic chemists that could help figure out, maybe you don't make it in algae anymore, right? Or do you build out you're actually growing algae at an industrial scale to isolate it. So there's a lot of different angles you could take that.
Just as a follow-up, so will that center, do you envision that center serving as incubator site with the infrastructure to like kind of enable these potential startups to grow?
One way, do you want to take it?
No.
I mean, one way to take is, can we attract a large pharmaceutical company? They know how to do this, right? And so it's not that we're starting a company, it is that we are licensing something to a company.
But the same thing, and we have experience with that, large pharmaceutical companies, once they come across the bridge into Maine, they look for people. Even a large company, they have to find people living in Maine. They're not bringing the people from Denmark or from France or from other states in the US here.
We could if we had better immigration.
And we don't have this. So the workforce training is very important. And we can talk about that. We are thinking at the moment about a curriculum, a year-long curriculum, where you actually go from basic research all the way how to negotiate with the FDA about a compound you have found, making a business plan in between. And we are not doing this, we're too small for an incubator at MDIBL, but we can do this with our students. We can do this as an educational effort. And this is what we're thinking about at the moment.
And I'll just maybe add one more thing to your question, right? This is very early stages. So imagining what it's going to look like if it grows into an incubator, the sky's the limit. But I think for one of the things I'm excited about, right, there's a lot of enthusiasm in the state for trying to think about this, right? Even here in Portland with ocean cluster and things like that, right? So a lot of people are thinking in this space of what does innovation in the blue economy and biotech look like for our state. And the good thing is we're talking to each other. So to be determined what it ends up looking like. But I think the enthusiasm is there to try to solve the problems of, bringing the workforce and keeping them here and helping them find housing and all those things.
So Beth, to the point you made, and Jim, oh, sorry, do we have that?
Good question.
Yeah, go ahead. Oh, sorry, go ahead.
Just a related question, both of you, Dr. Holler as well as Jim. What's the point where you actually get involvement through government bills or politically? Because obviously that whole system could really jumpstart this whole program through grants, through initiatives, through whatever.
The fascinating thing at the moment is that our collaboration and what we need for that is relatively small. So that's really the fascinating, and you can feel this, hopefully, the fascination of this project, because it really, as you realize first, there are two partners coming together and we can start right away. And then once we have compounds, then we need a lot of support. And it may well be that in between, when we try to isolate these compounds, then we need structural chemists, and then we need a lot of other people. And then all of a sudden, even the characterization of these compounds becomes something where we need more support. But the wonderful thing initially is that we will start with what we have at the moment. I mean, this will be, sure, we need resources from the individual institutions, but the fascination is there, and we will see whether we have something, and then we go from there.
And the thing about oceanography, it's such a collaborative discipline because you can't do everything on a ship in one, right? So you just like we found the partner, we'll find the next partner we need to fill that gap.
To your specific point, Lou, I think, the state has done a good job of creating resources like the Maine Technology Institute that often can kind of provide seed funding to get some of these initiatives off the ground. But I do think there is a tremendous opportunity for the legislature to, continue to invest in these kinds of activities. I know there's a $100 million bond that's still sort of nascent there, waiting to be, taken up again in this second session. So those kinds of investments are really critical to creating the ecosystem that we've really been talking about here, right? So attracting the workforce, attracting the venture capital folks.
I would go even
attracting-
far, I would go take a step back even further, though. You can attract a workforce, but if there's no place to live, that's a problem, right? And we-
that's why we get, the houses involved.
Right, that's where you've got to get legislation involved. We've got to solve the housing crisis in the state of Maine.
It's all related to the same issue.
Right, and immigration, I mean, the United States was where the best and the brightest of the entire world wanted to come. And we are making it harder and harder and harder for people to come, highly educated people with the skill sets we need. And that's how you can jumpstart. So I think immigration is a huge part, I think, of how Maine could solve a number of problems quick. But then again, they got to have a place to live.
Right.
So the housing, it really comes back to housing for me in a lot of these issues.
Yeah.
So with immigration, small thing at MDIBL, but I personally believe it's the individual person. So we have now graduate program, graduate student program at MDIBL, international graduate student program, where we have 25 at the moment. And when I see that some of them can even think about staying in Maine after their 3 to 4 year education, because there are projects and this is interesting. This is how I think we can start.
And I'll just add one more thing, that we're lucky in the state of Maine that, our legislatures, they support science. They care about what we do. We just had a groundbreaking at Bigelow on Thursday for a new center for education and innovation. And, had representation, Senator Collins was there with a shovel, helping us build this new building. They want to support.
Seriously, with a shovel. The woman
Yeah.
she's done a lot of groundbreakings. She,
Right, so I think that we are well poised, right? When we come to our delegations and say, hey, here's a growth opportunity for our state. How can you help us with this? Whether it be through bricks and mortar or supporting other programs to enable us to bring the workforce here. I think that's a unique position that we have here in Maine.
Thank you.
But we talked earlier about collaborations, and I think this is really very important to realize that. And it happens a lot in Maine, but nonetheless, we have such a lot of very talented institutions. We have been, that's not Maine, but New Hampshire, but we have talked to Army about regeneration, lots of activities going on. But coming back to this, our collaboration is really not something which normally happens. It's really like a marriage in heaven, because we can use these animals, and I'm really very optimistic that we'll find fascinating new substances.
It's very exciting. So a couple of comments have sort of centered around timing. Jim, can you just lay out what you hope or envision the timeframe for this project, and kind of what are the big benchmarks or milestones that you'll be looking at?
Sure. So I think we've been actively discussing the timing of the pilot project. We hope to sort of validate our animal models and the use of our animal models using a handful of algae strains to begin with, and then that process and sort of getting everybody through that initial screening will be probably 3 or 4 months. And then from that point, we're really ready to scale up and move into a more full screening program. There are 3,500 different strains.
Yeah, more or less.
it's a little
Yeah.
more.
Yeah.
So you can imagine going from a handful to that number of strains is a significant undertaking. So I imagine what we're trying to do is to look at how we triage that enormous library of strains to become to look at the most promising strains first and then move on from there. But we'll learn a lot through the pilot phase of the program, and I think that'll inform where we go from here. But it's certainly going to take support both from a financial perspective and from just the organizations coming together to make it happen. And so we're excited about where this can go, but got to walk before you run. So
That's great. Well, a huge thank you to all of you. This is
Well-
incredibly exciting. Yeah, Dr. Heller, always.
He's not letting you go yet.
No. I wouldn't. I mean, first of all, I think that's the time course we're thinking about, and but the question some of you may have thought of, and then you were too polite to ask, is, I mean, if this is so wonderful and so exciting, why is it not already happening somewhere else? I mean, do you think you are the brightest here and nobody has had these ideas before? And I can tell you, yes, we are the brightest. Because there are two reasons for that. One is nobody has this amount of differentiality. I mean, this is where you said, oh, in the beginning. And at the moment, nobody has these tiny animals, the engineered animals we are using for this approach. Tim and I, we have been talking to the pharmaceutical industry, and every time they hear us talk, they say, oh, great, wonderful, we should use these as well. And then they go back home, big companies, and they sit around the table and they said, okay, we'll try it eventually and all that. But now we can do it ourselves. We can use our wonderful animal models ourselves. So it really is very novel that this is coming together. And we are right.
You heard it here first. Well, this is really great and exciting, a wonderful thing for Maine. I think it's something that we really look forward to keeping you informed of. I hope you will continue to stay tuned. As I mentioned, we'll get some news out about this probably in the coming week or so. And then if you're interested, you can sign up on our website. There is a little form out there if you want to receive our updates about this and other projects happening at MDIBL and around the state. And we are thrilled to have you all here with us this evening. Thank you so much for joining us. And please, there are a lot of refreshments out there. Our panelists will stick around for continued conversation. And thanks so much for tuning in. We appreciate it.